Hello, biology students. I just want to take a little time to go through the lab that you did, the gelatin lab that we did last week, just to help you a little bit with some of the calculations and understanding exactly what's expected and what we're supposed to learn from this particular lab. So we had three, and it says gelatin cell diffusion on your lab sheet. So we know that we're using gelatin to simulate what happens with cells, which are, it's kind of difficult for us to see this at the cellular level. So we're using the gelatin as that model. So things enter the cell through the membrane by simple and then diffuse into the cell and into toward the center of the cell and around in the cell. And that's how the cell actually gets the nutrition that it needs to function. So we're really looking at that model. So on your sheet, and I apologize for the messy writing, I just threw this together really quickly. So we had three cubes that were 1 centimeter, 2 centimeters, and 3 centimeters on each side. And on the sheet, it asks you to convert those to millimeters, which we said 1 centimeter is 10 millimeters, and then we have 20 millimeters and 30 millimeters and so on. And we talked already about, okay, so there's 6 sides. So you have to calculate the surface area of a side by taking the length times the width, which would be 10 times 10, but then multiplying that by 6 because we have 6 sides. Volume is length times width times height, and since we have a cube, it's 10 times 10, which is 100, times 10, which is 1,000 millimeters cubed. And then surface area to volume ratio is really taking that surface area divided by the volume. And we want to express this as a ratio. So we want the surface area to volume, I would like you to express that as a ratio, not a fraction. So if I take 600 divided by 1,000, it is 0.6, and that's a 0.6 to 1 ratio. That's what our ratio is. So 0.6 of cubic millimeters of surface area. Sorry, that's squared millimeters to 1 cubic millimeter of volume. So it's 0.6 to 1. Now you have to do the same calculations with 20 millimeters. So, right, 20 times 20 times 6 sides, right? So we're doing the same thing only with 20 and then the same thing again with the 30. Distance penetrated. This is where we're not all going to have the same answer for this. I will say, regardless of whether, like some of you maybe found that it penetrated in 3 millimeters, some of you found that it penetrated in 5 millimeters. This can be a little bit tricky because we have to think about, we have this cube of gelatin. So let's say that this is the, let me start over here. Let's say that I have a 30 millimeter cubic gelatin cube. And let's say that I cut this apart and I measured with a ruler how far in this actually penetrated. So it's going to do I'm going to say that mine penetrated in 3. I measure the width of this. And let's say that it penetrated in 3 millimeters. So on your sheet, it asks for the distance penetrated. For mine, I'm going to say 3. Everybody's is going to be different. Please, I don't just write 3 because I said 3. I think that's, please, I hope you did your own measurements. And so distance penetrated is 3 millimeters. The distance not penetrated then, so here's here's where I want to make sure we have a good understanding. If it penetrated in 3 millimeters on this side, it also penetrated in 3 millimeters on this side. So if I take 30 minus these 3 minus these 3, the distance not penetrated is going to be 24. Sorry, writing with a mouse, super messy. So for me, the distance not penetrated, oh, I'm going to go down to my, because I was doing the 30. The distance not penetrated in my 30 millimeter cube is 24 millimeters. The next column then is volume not penetrated. So just like you found the volume of your original cube, I have to erase this because I had some data in here from before. So how am I going to find the volume not penetrated? Well, I'm going to take this size of cube, 24 times 24 times 24. That will give me the volume not penetrated. And you have to do that with each of these. So, for example, if you saw that your 10 millimeter cube, that liquid penetrated all the way through, then I have 0 distance not penetrated. And maybe I measure on this, and maybe I had, and I'm just pretending here, maybe I had 5 millimeters penetrated, so I have just a small little cube in the center that wasn't penetrated. And maybe for this one, I really can't tell, but if the other ones were 5, these should all be 5. The distance that it penetrated in one cube should be the distance that it penetrated in all the cubes. So you really probably only have to measure one, and I would pick the largest one, and then I would check and see, did it penetrate all the way to the center with the 10 millimeter one? And I don't know, I didn't actually see all of your results, so I'm not sure what you got. But this calculation is based on what you measured, but it should be the same for all 3 of your cubes. And then the distance penetrated, again, is going to be this subtracted from both sides of your cube if you measure it across. So if I, of course, penetrated in 5 millimeters on my 20 millimeter cube, I have to take this off of both sides. So I have 10, right? I would have 10 millimeters not penetrated. And this, of course, would be different if it was 5 as well. This would actually be 20. So I don't want you to copy my answers. I want you to copy my methods here so that you know how to calculate this. All right, volume not penetrated. Again, we're going to take that inner cube that's not penetrated and so if this was 10, it'd be 10 times 10 times 10. That would be the volume not penetrated. And then the percent, and I don't have that column on here, but the percent total volume not penetrated. Now I'm going to calculate the percent of what was not penetrated compared to my original size of the cube. So if you can follow this, I'm going to take my original for this very last column, which again, I didn't fit it on here, I apologize. But percent of volume not penetrated, I'm going to take, let's start with the 30. If the distance not penetrated was 20 cubic millimeters, I'm going to divide that by 30, and that's going to be times 100, and that will give me my percent that's not penetrated. So again, I would take my volume not penetrated. Oh, just kidding. No, I wouldn't take 20. I would take this answer. Volume not penetrated divided by original volume, okay? Volume not penetrated divided by original volume times 100 to get it in percent. And then that's your answer for percent of the volume not penetrated. And hopefully you can look at your data when you're all done and come up with some conclusions. They've had some of the questions like, you know, why, how does the surface area to volume ratio and the percent of total volume not penetrated vary as the cube size increases? how small must a gelatin cube be based on your results for the solution to completely penetrate in 15 minutes? So answer those questions. And that should be it. And you can ask me questions, I think on Wednesday when you come. I think that would be totally fine if you have a couple questions on that. And then I want to talk a little bit about the results of the other lab. I know we kind of ran out of time. I didn't want to take another day in class to let you kind of work through the rest of that nutrient testing lab, just because when you guys are here, I really do want to cram as much information as possible. I want to do as many labs as we possibly can while we're here. And so I just didn't want to take another day to kind of go through this. So I did this for you, and I'm going to kind of show you the results. So for the lipid test, I said on brown paper. So here's what I did. I put the unknown on, whoops, mirror image, sorry. I put the unknown and then also oil and then water. So oil is my positive control. We know that oil is a lipid, right? Because lipids are oils and fats. That's what they are. So we know we would expect that the oil is a positive lipid test. And so the oil, you can see, I'm going to move really close here. I'll try not to get my face really close. Scary. But you see this discoloration. You know how it is when you get something greasy. Oops, that's the valve. When you get something greasy even on your shirt, right? So I don't know if any of you do your own laundry, but if I see that in a shirt, I usually pre-treat it a little bit because it's a little difficult to get out, but it leaves this oil stain in your clothes, right? So maybe you're frying bacon or whatever you're doing. So this is my positive test. Water is obviously the negative test. You can't even tell any water was put on here, right? It dried and it's made no difference to the brown paper. My unknown, and I had to put tape on here. I'm going to put it really close. I had to put tape on here because it dried and cracked, but you can see there's no discoloration of the paper. So this is really a negative test for lipids, okay? This just dried and cracked off, and definitely not a positive test for lipids. So I don't think any of you got to that point. And it's a really quick, easy, and it kind of really has to sit until the water and this unknown dries anyway. So it would have had to sit overnight for you guys. So hopefully that's the last piece. We can, if you guys want to do a Google Meet and kind of talk about the rest of the results, if you want to do that, we could. All right, so then I wanted to take just a couple of minutes to go through the lipid test for your nutrient testing lab, which I know you didn't get to do. I don't think anybody got to that, and that's okay. There's no special technique involved, and I don't want to take another day on this particular lab because I'd rather move on and have you do another lab. I want you to have as many lab experiences as possible while you're here in class, and then the other stuff can be done outside of class. So here's my results. I put some oil. I'll try not to get my face too much in there. Some water, and then our unknown, which you can see, whoops, dried and cracked. I had to tape it on here. But you can see there's no lipid stain left here. This is what we would expect, like oils kind of stain or leave that oily residue that doesn't really dry. Water, so oil was our positive control, water was our negative control, and then the unknown we were testing for lipids, and I would say this is a negative test. I don't see any oily residue left on here. So that is the information on our lipid test. And I'm just going to zip through some results really quickly. For our test for proteins, the unknown should have tested positive for proteins, as should have the albumin and the pepsin. All other things should have been negative for protein. For starch, the onion, the potato, and the starch suspension should have been the only three that tested positive. For the glucose, and you might have seen a slight positive result in the unknown. And then for lipids, we said the unknown does not contain any lipid. The unknown was fat-free yogurt. So I know there's not any fat in there, but there is some sugars.